Two men, identical twins, same DNA, same parents, same bedroom growing up. At age 55, a researcher ran a molecular assay on both of them, a test that reads specific chemical marks sitting directly on top of their DNA to calculate exactly how fast their biology is aging. The result: one twin had a biological age of 52, the other 63. Same DNA, 11 years apart on the biological clock. The difference had absolutely nothing to do with the hand they were dealt. It was entirely explained by the environment their genes were listening to. And right now, your genes are listening to you. If you are over 40, your daily habits have already picked a trajectory, and you might be aging a decade faster than your calendar without even knowing it. In this video, we are going to completely unmask the hidden environmental commands that flip that biological switch, so you can force your system into a state of renewal. For most of the 20th century, the medical narrative was simple, rigid, and wrong. Your genes are your destiny. You inherit a blueprint, and that blueprint determines what gets built. If your father had heart disease, watch out. If your mother had diabetes, you are probably next. You play the cards you are dealt. That story is fundamentally incomplete. Every single cell in your body carries identical DNA. A liver cell, a neuron in your brain, and a muscle fiber in your leg all hold the exact same genetic code. Yet they behave completely different. They build different structures. They perform entirely different jobs. Something sits above the blueprint, deciding which pages get read and which pages stay permanently silent. That something is the third and final system controlling your rate of aging, the control room. Science calls this epigenetics, from the Greek, above the genome, the molecular software layer that dictates which genes switch on and which ones remain quiet. And here is the truth that changes everything you've been told about your longevity. That software layer is rewriting itself in response to your life right now. To understand how deep this runs, we have to look backward. In the winter of 1944, a brutal Nazi blockade in the Western Netherlands created a catastrophic famine known as the Dutch Hunger Winter. Women pregnant during this famine gave birth to children who, decades later in peacetime, well-fed, affluent, and well-housed, suffered from unprecedented rates of obesity, cardiovascular disease, and metabolic dysfunction. But the story doesn't stop there. Researchers eventually looked at the grandchildren of those pregnant women. The exact same metabolic vulnerabilities appeared. The grandmother's starvation had altered the epigenetic software marks on her eggs. The experience itself wasn't transferred, but the molecular signature of that experience was inherited across generations. Transition back to the present. Your genes are carrying instructions written before you were born, and the choices you make before noon tomorrow will write instructions into your DNA that can outlast you. The bad news is you can inherit a stacked deck. The incredible news is you are holding the pen. Here's how the control room actually writes those instructions into your biology. It uses two elegant primary mechanisms. The first is DNA methylation. Think of a small chemical tag, a methyl group, attaching to a gene like a Post-it note stuck over a paragraph in a book. The paragraph is still printed there, but it gets skipped. The gene exists, but it becomes a silent ghost to the rest of your genome. The second is histone modification. Your DNA wraps around protein spools called histones. When the wrap is tight, the gene is locked away, inaccessible. When it loosens, the gene can be read and expressed. Specific enzymes tighten and loosen these spools continuously, acting like volume sliders for your genetic output. These marks are not permanent. They are added, removed, slid up, and dialed down every single day based on the molecular instructions arriving directly from your environment. We have already mapped how your habits affect the power grid and the architecture. But the control room reads those exact same inputs through a totally different lens. It views them as a real-time broadcast. Every single morning, you are sitting in a live gene editing session. When you initiate purposeful physical demand, specifically sustained aerobic effort or progressive resistance training, your body doesn't just burn fuel. The mechanical stress forces your cells to instantly demethylate specific metabolic genes. You are literally lifting the chemical tags that suppress your mitochondrial repair machinery. When you encounter controlled thermal stress, the control room responds immediately. Intense cold activates the expression of brown adipose tissue genes to flip your metabolic thermostat. Intense heat activates heat shock proteins through direct genomic signaling, silencing entire clusters of pro-inflammatory genes before they can ever cause damage. Your diet is literally the ink the control room uses to write. Micronutrients like folate provide the raw materials your methylation cycle requires to function. Compounds like sulforaphane, found in cruciferous vegetables, block the specific enzymes that keep your tumor suppressor genes tightly wound. Eating is not consumption. It is an informational download into your genome. During deep, slow-wave sleep, the control room runs its master diagnostic cleanup. This is the precise window where accumulated methylation errors from your waking hours are corrected, and inflammatory gene expressions are dialed back down to zero. If your sleep architecture is fractured by electromagnetic alerts or poor hygiene, the correction window stays locked. Perhaps the most potent writer in the control room is your nervous system state. Chronic, unresolved psychological stress methylates a critical gene called NR3C1. This specific gene governs your cortisol receptors. When it is chronically methylated, your receptors lose sensitivity, your stress-off switch breaks, and cortisol runs unchecked. What you experience as chronic, unexplained anxiety often has a literal molecular address in your epigenome. Even human connection changes the data. Loneliness activates inflammatory gene clusters, while an actual felt sense of purpose sends an explicit signal that stabilizes genomic structure. If this sounds like elegant theory, look at the hard data. In 2008, Dr. Dean Ornish took a group of men with low-risk prostate cancer and put them through a comprehensive lifestyle intervention. 3 months. Clean diet, targeted movement, stress management, and community support. No drugs, no surgeries, no clinical procedures. After just 12 weeks, they biopsied the tissue and ran a comprehensive gene expression analysis. Over 500 genes had completely changed their behavior. Tumor-promoting oncogenes had powered down, tumor-suppressing genes had aggressively activated. 12 weeks, 500 genes, changed entirely by a shift in the signal environment. Your DNA is not a fixed destiny. It is a listening device, the most sophisticated, sensitive, responsive interface in human biology. And it is taking its cues from the pattern you deliver every single day. If you calibrate the power grid, you give the body the energy to heal. If you stabilize the architecture, you stop the silent burn of inflammaging. And if you command the control room, you unlock the optimal expression of your genetic code. But there is a catch. These 3 systems do not operate in a vacuum. They are locked in a continuous compounding loop, and sitting right at the center of that loop is a single overlooked biological variable that acts as the master throttle. If this master throttle is miscalibrated, it will drag the power grid, the architecture, and the control room down with it, no matter how many supplements you take or how hard you train. But if you master this single variable, you gain complete operational command over your biological trajectory. That master throttle and the tactical field guide to controlling it is the single most critical piece of this entire journey. We'll go there in our very next video.